So the next presenter will be honored to have Dr. Mehta, right?
Parag Mehta.
Mehta.
Parag Mehta.
And he will present a product. I think a few years ago, I saw the news, a talk about the UCLA Medical School research team about head and the neck cancer. And you know, today we're so privileged to have you and take on that.
Thank you, and thank you very much, Roger, for inviting me yesterday to make a presentation here. I'm happy to be here in this company. How do I make a presentation? I should be able to share my screen, right?
Yeah, you can share your screen and then talk through your presentation.
Yeah, let me do that. Can everyone see my PowerPoint?
It's coming up. It's set as okay. Yeah, we see it.
Okay, give me a second. So I think, as I said, good afternoon, good evening, good morning, everyone. I'd love to talk to about our platform we have designed around the botanical drug origins. And I'll talk about all the assets we have, but we particularly focus on head and neck cancer, which is our lead program today. Since we are a Delaware C corporation, we will be raising capital, so there may be investors in the audience. So I would like to read out the safe harbor statement. Anything I say, you should do your own diligence before making an investment if you are considering. So let me give you an executive summary. So we are focused on orphan cancers right now. That's what I'm going to talk about today. But we also have a couple of other programs in a phase 2 ready asset for osteoporosis and osteoarthritis, which I will not talk about today, but just to let you know. Today we will talk about head and neck cancer and brain cancer. There are multi-billion-dollar opportunities. Both of them are highly unmet needs. And there are drugs, all right, but there are no FDA-approved drugs. They actually give a durable clinical response. And most of the people do not last very long on those drugs. We have taken a very different approach when we started a company. We ourselves are scientists and physicians and entrepreneurs, but I took a very different approach. Instead of building up a huge team, we partner with a number of major schools for basic research as well as for clinical trials. And you can see the list. We are working with a number of those universities, as someone just mentioned, UCLA Medical School, Stanford, Harvard, Barrow, and the US Department of Veteran Affairs, University of Miami, Mayo Clinic, Nebraska, University of Nebraska, and most recently, University of Texas Medical School. We are focusing on IP, and we have some drugs in a various stage of prosecution. One drug is one patent is already approved. And our drug, our head and neck cancer drug, I believe is the first botanical drug to receive the orphan drug designation from FDA. And I believe it is fourth or fifth in the history of FDA to receive that one. We are currently looking about $10 million right now, and followed by two tranches of about 20 million or thereabouts each. And idea is to go make a public offering in about 24 months. And we'll also submit the NDA for a lead program within 24 months. And that will lead us to additional 2 programs in pivotal trials. So something about a company, we are based in Bedford, Massachusetts. We have created a very different platform. Most people typically take a single molecule from a plant or they get extract, but the way we are creating drug is quite different. I'll talk about it a little bit. And they are actually a design combination of molecules. And our goal is to be really a powerhouse for our next generation of botanical drugs. People ask me, what do we want to be when we grow up? And my simple answer is, maybe a bit of a hubris on our part, but there is no reason to believe that the botanical drug company cannot be the next big multi-billion-dollar pharma company. And because I think it is calling upon us that we need to do this, because there's opportunity there and a promise of the drugs to bring it to the patients. So far, company has been funded by the management. We have an interesting investor syndicate, number of grants from the foundations, and some of our development partners put up some capital. Briefly, the team background, myself, I'm not a career pharma guy, but I'm a serial entrepreneur. This is my third company. In past, I've worked in different companies, but I happen to have a PhD in organic chemistry. That's kind of helpful in this drug business. Louis Avila is our VP of R&D. He's a career pharma guy. Right after finishing his postdoc at Harvard, he joined Focal and Genzyme and Sanofi. Bharat Tiwari, our board member, is a senior executive from pharma. He was a EVP at UCB and also SVP at Merck Serono. Guha is not in a pharma, but he understands chemistry well, and he is involved in the healthcare business. Otherwise, he's on a board of a hospital. I'm very proud of our advisory and clinical partnership board that we have assembled. These are all people who are extremely active in supporting various programs. I'll talk about that in a second. Nicole Shonka in Buffett Cancer Center and June Hom at Mayo, they are a PI in one of our drugs that we are doing for glioblastoma. Ikokobi Fongkam is also a PI on one of the other drug for glioblastoma, along with Joshua Levy in Texas, Chirag Patel at MD Anderson. And then we have Marilyn Van, Dr. Srivatsan at UCLA, and Elizabeth Franzman in UC Miami. They are working on their PIs on our lead program, which is in phase 2 for head and neck cancer. Eugene Shakhnovich and Daniel Hartle at Harvard, we have done some foundational work with them on showing why the multi-agent drugs are able to prevent the resistance in cancer. And John Kaminis is just another super guy, significant experience in pharma, good friend of mine, and as a very thoughtful advice for me. So when you talk about cancer, I think first, if you can back up for a second and think about why the cancer, despite all the money and effort devoted to cancer, why the burden is rising. And I think there was a wonderful essay in the Wall Street Journal some time back. I think that sums it up pretty neatly. So Asra Raza at Columbia, she said that, first of all, we have to agree that whatever we're doing in cancer is all wrong. We have to come off our high horse and admit that. And her point was very, 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 very specific. We are going after a single agent drug, going after simple genetic mutation, but cancer is indeed way too complex to be able to reduce to such reductionism. And therefore, Jason Schelzer, when published the papers in 2019, it made it to New York Times. And he said, therefore, in cancer, 97% of targeted therapies fail in the clinic. Despite that, people keep doing the same thing again and again, and they get the same results. And it's up to us, behooves us, to break new ground. So why does that happen? See, if you look at this particular slide, on the left side, you see the tumor. Tumor is molecularly very heterogeneous. What typical single agent drug does, it only attacks on one particular kind of tumor cell. So what happens is that you get early response, but then under the evolutionary pressure, first of all, the sensitive tumor becomes resistant, it mutates, and the tumor cells which were not originally addressed, they start growing. And so therefore, what happens is you have therapeutic failure. Now compare that with our plant-based drugs, and our plant-based drugs, and I'll explain in a second. What they do is, first of all, there are a number of molecules. Their ability to address tumor heterogeneity is much, much higher. Secondly, we are giving them in all the individual molecules on a low dose. And it's an amplification effect that you get by combination of those molecules. So what happens is that because each molecule is in a low dose, the tumor itself never faces evolutionary pressure to mutate. And therefore, the trajectory remains very as a drug-sensitive cell. Other important part in a tumor is that everybody focuses on a cancer cell, but we also look at the healthy cells, because the idea is that we have to rethink the whole, we don't have to annihilate every last cancer cell. Even if the few cancer cells are around, but you surround them by a significant number of healthy cells, which botanical drugs are very good at doing. If you do it, you arrest the cancer. And at worst, you make cancer like a diabetes or a heart disease. It's a chronic disease, but a person doesn't die from that. So that is our idea that we have translated into practice. And I think this was another interesting quote from Cassandra Kay at Emory. She said that if you look at my reading of her write-up is the following. Cancer at the end of the day is a biological problem. And there are many biological problems in nature, but nature solves the biological problem. But it does not solve by finding a silver bullet molecule. What it relies on many more compounds and interaction among them to solve the problem. And that's exactly what botanical drugs do. So what we do is we take these molecules, which are orthologous to human endogenous metabolites. Many of you here know probably that look at cellular machinery of the plant cell and human cell. The most of the proteins and enzymes are orthologous, meaning that there are the functional equivalent of that in a plant. So therefore, when plant molecules, when you look at that and do a structure comparison with the endogenous metabolite the body produces, they are significantly similar. So there is the idea that if you pick the molecules which are similar to human endogenous metabolites, it solves a number of problems that synthetic drugs have. You don't hit a metabolic dead wall. Yeah, you don't have a problem binding to the proteins, and you are able to counteract the higher order effects among the various mutated genes. So we are creating drugs like that, and we believe that I think these drugs can be developed at a much lower cost than typical synthetic modalities. All our drugs are administered orally. We have, as a patented by, protected by the composition matter. And I think one of the things that many people in today's conference might be aware of it, but many people in other in pharma companies not aware the primary precedent that FDA has, and that is even better than any kind of patent protection you can get. So we have four INDs approved. Actually, five. Fifth one I'm not shown here, but the fifth one is approved for graft-versus-host disease. And there is one more. We have a phase 2 ready asset for osteoporosis. We are working with 4 different plants, but the one I'm going to talk about is APG-157 program number 1 that we are in UCLA and Miami. Second one is also for a combining with Keytruda. First such a combination IND approved by FDA for combining with Keytruda. And then third and fourth that we are doing in a different cohorts of the glioma patients at Mayo and Nebraska and Baylor Scott and White in Texas. So let me walk you through our strategy, how we develop this particular drug. When you look at the head and neck cancer, what is currently going on, it's a pretty bad cancer. In US, 1 person dies every hour. And in India, it's every 10 minutes. I personally lost 5 people to head and neck cancer, so it's very close to my heart. If you look at it right now, most of the drugs are old drugs. The EGFR inhibitor, cetuximab, but barely works. Even the latest and the greatest checkpoint inhibitors have a very poor response. So essentially, all these drugs are generally only applicable at stage 3 and 4 or a metastatic patient. But for early stage, there are no drugs. And so we think that there is a great opportunity. There's essentially a wide field opportunity to play in a number of different areas within head and neck cancer. And so that's what we focused on. So if you look at the head and neck cancer and look at the signaling pathways, it's fairly complex. To assume that we can go only after EGFR on the left side of the screen and we can inhibit that and stop the tumor, that is not a right thinking. And therefore, all these drugs generally fail to provide a durable response. So what we are doing is, and so if you look at the current drugs, they do one thing. They focus only on one thing, causing the cell death. And either chemotherapy or IO drugs or EGFR inhibitor. That's all they do. But what we are saying is that alone is not sufficient. If you really want to cure cancer, you have to think differently. And this is how we think. So what we do is our drugs also cause the death of the cancer, but very selective apoptosis of cancer of the cancer cells, and only focus on a BCL2 class of protein, just like a drug from AbbVie does. We also modulate NF-kappa B and P53. And we also stimulate the immune system by recruiting a TILs to the tumor. And those together cause the death of the cancer cell. But it's also important to prevent the progression of the cancer. It's not sufficient to just kill the cancer cells. And I think to do that, we have to look at it very differently. Chronic inflammation, and the first time we are looking at oral microbiome, because that plays a big role in this cancer. And oral microbiome or microbiome in general, I think is a fool's errand when people think they can find a single molecule drug and deal with that. It cannot be reduced to molecular target. And more importantly, in head and neck cancer, the recurrence rate is very, very high. So we have to prevent the recurrence if you want to deliver response. And so EMI1 cancer stem cell play a key role in this one. And our drug also prevents or rather arrests the cancer stem cells. So drug is from a well-known plant. People here should know that, curcuma longa. And curcumin, everybody knows what curcumin is. There are million supplements with curcumin around. But curcumin is a wonderful molecule, but that's not the only molecule the plant has. It has 230 different molecules. And what we have done is we have taken very specific molecules and create our combination, rational combination. And they generally fall in 3 categories. There are non-flavonoid small molecules, there are flavonoids, and there are some macromolecules. And this is the first time we have created a drug that, so head and neck cancer, all the drugs that I mentioned, they are given IV. And despite the fact that oral cancer is one of the most accessible cancer after after the skin cancer. So we designed the drug in such a way that drug is encased in a pastille that slowly dissolves in your mouth, and drug gets directly absorbed in a tumor, as well as it gets into a systemic absorption by sublingual absorption. So it's a very unique drug, and very easy for patient to take it. So I'll walk through some preclinical results and then our phase 1 results and talk about our phase 2. So I won't belabor the point, but we have done enormous amount of preclinical work like anybody else would do in typical in vitro work, as well as new syngeneic mice xenograft. And I'll show some of the data right now. But suffice to say that we convinced ourselves that drug is, when we designed the drug by combining multiple molecules, including a lot of sequencing, and that that drug works. So that to convince ourselves in the preclinical work that drug really works. One interesting thing I want to talk about that. So I don't know if you know, but BCL2 class of protein, BCL2 inhibitor is the only apoptosis inhibitor approved by FDA. And Bob Horowitz at MIT got a Nobel Prize for it. I had a fascinating discussion with Bob on the drug that we have. And he also recognizes that if you only focus on BCL2, very quickly cancer is going to mutate and develop resistance. Ours is the only drug that simultaneously downregulates all the anti-apoptotic genes and upregulates all the pro-apoptotic genes. So this was not lost on Bob either, and he saw that, and he was quite pleased that we are doing that. So it's a very unique drug, and this kind of a thing is only possible with a botanical drug. Interestingly, we took our drug APG-157 and we did syngeneic mice. We did two interesting things. We have done a lot of studies, but I'm showing you one. When you look at a control and on the right side, the mice that got drug, yeah, we reduced the tumor and volume and weight and all that. But very interestingly, that we increased the recruitment of CD8 positive T cells in the tumor by giving the drug. And CD8 positive T cells, right from a COVID to everything else, they are some of the best T cells that you want to have to protect yourself and to fight off the disease. And our drug was able to do that. I'll show you soon that it also does in human. And so one of the interesting things we did is, in addition to CD8, CD3 is a very important another TIL also. And as far as we know, both these lymphocytes are positively correlated with head and neck survival. And so if we can increase that, that is great. And we were able to do that. These are, as far as we know, this is the only drug that can do can upregulate both of them at the same time. Another interesting thing is that people talk about microbiome quite a bit. In particular, in cancer these days, when people are looking at the checkpoint inhibitors, it is found that checkpoint inhibitors work much better when there is a certain species are present in a microbiome of a patient. So what we did, we actually looked into in the mouse microbiome during the study and found that this particular species, Akkermansia and Lectospiraceae, which actually positively correlate with the effect of the immune system, by giving the drug, we were able to upregulate them. So this was just a data point in a mouse, but still very useful data point. So let me talk about this unique drug that we got IND approved. We have not started this particular trial yet, but I'll talk about it. So when checkpoint inhibitors work, one of the proxy for them in the in vitro study is interferon gamma. So you can see in this particular slide, the top two lines, blue and pink, interferon gamma does slow down the growth of tumor. But look at APG-157, the green and the brownish one that you see in the middle of the screen, they are much, much better than interferon gamma alone. And when you combine interferon gamma with APG-157, the effect is much better. But this was just a proxy to test for the actually in a living system how the drug will behave in combination with the checkpoint inhibitors. And then we actually did in the live animals the study. On the right side, you see that in the slide you can see that when you get checkpoint inhibitor by itself, NDPD1, very quickly tumor starts growing. APG-157 is much better than a checkpoint inhibitor, but when you combine both of them together, the results are significantly better. And this is very important because, and I think going back to earlier discussion we were having just before I started, one of the way I think we can convince big pharma is to show that where their drugs are falling short, and by combining with our drug, they can actually make their drug better. It's a Prius model. Instead of directly going to Tesla, why don't you start with Prius and establish the value, and then you can go with the and then you can develop your Tesla or a full-fledged botanical drug. So that was a part of the approach that we took in this study. Interestingly, what you see is, I think recruitment of CD4 and CD8 is very, very important for cancer. And you can see that on the right side, again, by combining both the drugs, CD4 and CD8 T cells significantly improve levels of them. So let me talk about our neoadjuvant therapy for head and neck cancer. Right now, there is no FDA-approved neoadjuvant therapy for head and neck cancer. In some cases, people use cisplatin and Taxol and all those kind of things, but they are very toxic and doctors don't like to use it, but when they don't have a choice, they use it. So we did a 25-person study at UCLA, actually the VA hospital in Los Angeles. We tested 2 different doses, 300 milligrams a day and 600 milligrams a day. No safety event whatsoever. And we look at so we are doing 2 things in the study in the endpoints. One of the endpoint was to look and to prove out our hypothesis that we had in a preclinical, that if you're actually blocking P53, upregulating P53 and blocking NF-kappa B to cause apoptosis, then the downstream cytokine should come down. And that we wanted to see. And that is a proxy for killing the tumor cells. We also wanted to see that what happens to CD8 positive T cells in actual patient. And we also wanted to first time test that in oral microbiome, are we able to reverse the dysbiosis of the oral microbiome? And we tested by 16S RNA sequencing, and we will do that. And then we did some other thing, which is very interesting, companion diagnostic test we came up with that using a cell-free RNA. And I'll talk about that too. So these are the patients. So if you look at this slide, right, on the green one is a placebo. In phase 2, we have no placebo. The phase 1, we had a placebo. Clearly, dose-dependent reduction, statistician told me that 25% reduction is considered significant, and we beat it, we hit it out of the ballpark. We did significantly more than 25% reduction of IL-8, IL-1 beta, and TNF-alpha, all 3 of them. So we are quite happy with that, and that shows that the tumor is breaking down. And I show another result why we believe the tumors are breaking down. This is a very interesting study. So we took a patient, this is a floor of the mouth stage 4 cancer. And if you look at the left side, before the drug was given, the biopsy was done, immunohistochemistry was done, there are hardly any T cells in the tumor tissue. On the right side, what you see in immunofluorescence, that after giving the drug, there are plethora of T cells infiltrating the tumor, which is very, very good. And so that's what our PIs are telling me, that it's very good, and they are quite excited about it. I'm not an immunohistochemist, so I can't explain more than that, but I think that's what I was told. Other interesting thing we did, that we are actually in a similar immune system. How do we know that? So apart from looking at the immunohistochemistry, what we did was that when tumor cell breaks down, all the debris of that one are picked up in a systemic circulation in blood. So what we did, we collected patients' blood, and we identified the cell-free RNA, which are involved in transcription of those proteins, and we find out the leukocyte activation is happening. And so that exactly is indirect, or rather direct explanation of the both system, immune system activation, also in the breakdown of the cancer cell. We also see the same thing specific to T cell. I can't explain this slide, but there are others in our company who can. And what I've been told is that, again, in the same test, the plethora of abundance of T cells are recruited to tumor. So that was done. Another interesting thing that we are looking into that, and we always know food as the medicine. And as I come from India, in many traditional system of medicine, they say that a microbiome, your stomach, everything. If your stomach is healthy, then you'll be disease free. And I think what they're referring to that in this traditional doctors talk about it, I think they're referring to microbiome. So a microbiome does not only start in the gut, it starts in the mouth. And there are fascinating papers from Taiwan and from NYU that said they correlated the changes in a bacterial level or a microbe level in a oral cancer patient, particularly bacteroidetes, livers increase, and Firmicutes decrease. So our therapeutic goal ought to be reversing that ratio. And that exactly the first, so everybody says you should do that, but nobody actually developed a drug to actually do that. And so what we did was we tested them. And on the left side, you see the placebo. Placebo has some effect, only because that you are disturbing the microbiome by putting the pest in your mouth. But look on the right side. By changing the Firmicute to bacteria ratio, I think 100 milligram 3 times a day is a significantly more than a 22%, 82%. But 200 milligrams 3 times a day as the highest change is able to affect. We have a lot more data on this one too, but this is just a very high level summary of that. And we are quite pleased with this result. So in our head and neck cancer phase 2, what we are doing right now is that typically when patient comes in, patient is stage TNM staging is given. And in US and in Europe, most places, the patient does not get any kind of definitive therapy for 4 to 8 weeks. And during that time, cancer grows by about 1.8% per day. So if you can imagine, in 4 weeks, by the time patient even gets around to getting any kind of treatment, cancer would have gone by 40, 50%. So and right now, patient is just getting sicker. So this is the first induction therapy that we came up with, where the goal is to stop the cancer right there and doesn't grow anymore, and better yet, reduce the tumor burden. So even if the patient undergoes a surgery at that point, the surgical resection is much smaller. And this is a very unique trial. Usually, as you know, in cancer, FDA only gives you approval in end-stage patient, the hopeless population or hospice population. Very rarely, FDA gives you approval for testing in a frontline setting. So when we did some study in a phase 1, we went back to FDA, and FDA gave us a frontline setting approval where this is given before any kind of a definitive therapy is given. And we are going with the same gold standard primary endpoint, objective response rate using a persistent RECIST. And we are also looking at a number of other secondary endpoints that I mentioned in a microbiome and a T cell recruitment. So we are doing that. And except with the exception of patients with the metastasis, all newly diagnosed patients are recruited in the trial. We have published extensively. I think that is another important thing that we need to get all of us need to get a more credibility that we just publish and at a mainstream conferences. So we have published in 2 major peer-reviewed journals, number of presentations we made in a society meetings. So we have done that. We have significant IP. So we have 4 patents. One is issued, 3 are in different stages of approval right now. We have a trade secrets like everyone else. And I think preventive precedent is a very, very powerful precedent. That drug was approved in 1941, a multi-agent drug, and FDA rejected every single generic application after that because nobody could actually figure out the exact composition except Pfizer. And so Pfizer continues to have a great run with that particular drug. As I said, we already have orphan drug designation. We already discussing fast track designation with the FDA on that one. And depending on data, we may go for a breakthrough therapy also. Finally, so we said the APG-157 is first botanical drug that is orally given and based on a very sound principle, evolutionary biology. It's just not that sufficient is a botanical in nature, but how we are putting the drug together is quite important. And as I said, as opposed to going after a single target or single phenomenon, it's going after apoptosis, modulates microbiome, stimulates the immune system by making it tumor hot, and then it works not only topically but also systemic absorption because there are also micromets in these patients. So it's a unique drug that we have developed. We have very good manufacturing and CMC. There are 2 companies I've worked with. They are top notch. They have been inspected by FDA, MHRA, DCGI, WHO, and so far so. I know there's some more work to be done in CMC before phase 3, but I think so far so we are good. Our goal is right now, I'm raising 10 million right now. I have some commitment right now. Planning to raise additional 40 million in a couple of tranches. One tranche would be great, but I think I'm being practical about it. Maybe a couple of tranches. And I think best way, I think in my previous life, I've done a public offering. And my feeling is that the companies like us, if we can do a public offering, I think we should do it because capital markets are wide and deep. And there is opportunity receptivity for all kinds of great ideas. I've spoken to a number of investment bankers, and I was told that the portfolio we have, the approach we have is quite unique. And under right conditions, if we can hit the street, it will be well received. And I think that is how I'm thinking right now. So this is what this amount of money would do. So 10 million will complete the phase 2A right now, which will allow us to file for breakthrough designation for a lead asset. Other drug that I didn't talk as much about it, but this is a very unique second one that you see, APG-157 with TMZ and TTF. That's a very unique approval for FDA. So FDA, you know, gives you approval for compassionate use for a patient, but generally only at one patient at a time. Very rarely FDA gives you approval for a group of patients at the same time. So we got a very unique approval for FDA for direct treatment of a recurrent glioblastoma patient. And so that we're doing it. The FDA told us that we'll give you this approval. You can treat 10 to 12 patients, come back to us with the data, and then we'll work with you to chart out a path how to get the drug approved. So that's a very unique approach we have taken. Number 3 is combined with the bevacizumab, Avastin. Avastin has a significant issue with in a glioma. It barely works. It's about 3 to 4 months it works, and then it stops. And we believe there's scientific rationale by combining our drug with Avastin. And so we are doing that with the Mayo Clinic and Buffett Cancer Center in Nebraska. And the last 2, we are still, we'll see when that will start. But I think one of them will definitely get underway. And the rest of the milestones that we think within important milestones that we can reach within next 12 to 24 months are listed on the right side. And for investors, I think we are feel pretty good. If we can do all these things, we can have a very good liquidity event within 24 months. So thank you, and I'm happy to answer any questions.
Thank you, Doctor, for very impressive presentations. And so is this product you developed by your research team, or you developed based on traditional medicine somewhere?
No, we developed ourselves, but we took the cues from a traditional medicine. But so we developed, our team developed it.
Okay.
And somewhat in conjunction with UCLA. Okay.
And right now, what's your relationship with UCLA School of Medicine? And
well, they are clinical partners. So there are these people have a joint appointment at VA as well as UCLA. We are working with both, and so they are our clinical partners. So they are conducting clinical trials right now. As far as the IP is concerned, we own all the rights to the product.
I see.
Yeah, I have a question. How much have you spent so far? How much have you raised thus far?
So we have not publicly declared it right now, but it's sub 10 million.
Okay, all right.
But you can imagine that these things, what we have done doesn't come cheap. So it was a fair bit of amount.
Yes, okay.
It's okay.
Great presentation.
Thank you. Anyone else? Any investors here? Anybody else wants to invest? You're looking for it. I'll have to close the round as soon as possible.
Yeah. And are you have a firm timeline starting phase 3 in your mind?
Phase 3, you mean?
Yeah. Or
so I think so we call pivotal trial, not necessarily phase 3, but pivotal trial phase 2B or phase 3. I'm thinking sometime 3rd quarter of next year.
Okay, thank you.
Mm-hmm.
Steve, I thought you said.
Oh yes, no. I was I think as an orphan drug, that's a very interesting path. That this product. Am I muted?
No, I can hear you.
Me? Okay, great. Yeah, I think the orphan drug path is a very interesting path that this product has taken. I think the head and neck cancers. I think that was a very astute focus in terms of how to position your drug for market success. And I'm going to look more closely at your milestones in terms of your capital requirements. But as far as I see from your slide, that's right in there with what you require at this stage. And we're looking to put together those type of funding packages. We're looking to put together a special purpose vehicle that can potentially fund up through phase 3 for 5 to 10 drugs simultaneously. And so your drug would absolutely fit that profile, especially given where you are in your clinical development, as well as some other products that we've seen here today as well. So it just underscores the need for us to put together that type of an investment vehicle in order to accelerate to market these products.
No, that's great, Steve. I have some ideas too, and I think I like to interact with you folks more, maybe offline. I have some ideas how we can not only fund our company but also other companies, how we can pull together some resources and how do we go about doing that. Some ideas I'd like to talk with you guys offline.
Indeed, we'll follow up. Thank you.
Thank you. Great.
Thank
Okay.
you.
Yeah, thank you so much. Any other questions? Then thank you, Doctor, so much for the time and the wonderful production
Thank you.
presentations. We'll follow up on this with you, at another time, later time.
Great. Thank you for inviting me.
Thank you.
Mhm.
Bye-bye.